Gene therapy is when DNA is introduced into a patient to treat a genetic disease. The new DNA usually contains a functioning gene to correct the effects of the disease-causing mutation. Gene therapy uses sections of DNA, usually genes, to treat or prevent disease. The DNA is carefully selected to correct the effect of a mutated gene that is causing disease. The techniques was first developed in 1972 but has so far had limited success in treating human diseases. Gene therapy may be a promising treatment option for some genetic diseases, including muscular dystrophy and cystic fibrosis. There are two different types of gene therapy, depending on which types of cells are treated. Somatic gene therapy, transfer of a section of DNA to any cell of the body that doesn't produce sperm or egg cells. Effects of gene therapy will not be passed on to the patient's children. Germline gene therapy, transfer of a section of DNA to cells that produce eggs or sperm. Effects of gene therapy will be passed on to the patient's children and subsequent generations. Gene therapy techniques. There are several techniques for carrying out gene therapy. These include gene augmentation therapy. This is used to treat diseases caused by a mutation that stops a gene from producing a functional product, such as proteins. This therapy adds DNA containing a functional version of the lost gene back into the cell. The new gene produces a functioning product at sufficient levels to replace the protein that was originally missing. For example, this can be used to treat loss of function disorders such as cystic fibrosis by introducing a functional copy of the gene to correct the disease. See illustration below. Gene inhibition therapy, suitable for the treatment of infectious diseases such as cancer and inherited disease caused by inappropriate gene activity. The aim is to introduce a gene whose product either inhibits the expression of another gene, interferes with the activity of the product of another gene. The basis of this therapy is to eliminate the activity of a gene that encourages the growth of disease-related cells. See image below. Killing of specific cells. Suitable for diseases such as cancer that can be treated by destroying certain groups of cells. The aim is to insert DNA into a diseased cell that causes that cell to die. This can be achieved in one or two ways. The inserted DNA contains a suicide gene that produces a highly toxic product which kills the diseased cell. The inserted DNA causes expression of a protein that marks the cells so that the diseased cells are attacked by the body's natural immune system. See image below. How is DNA transfer done? A section of DNA gene containing instructions for making a useful protein is packed within a vector, usually a virus, bacterium, or plasmid. The vector acts as a vehicle to carry the new DNA into the cells of a patient with a genetic disease. Once inside the cells of the patient, the DNA gene is expressed by the cell's normal machinery, leading to production of the therapeutic protein and treatment of the patient diseased. Challenges of gene therapy. Delivering the gene to the right place and switching it on. Delivering a gene into the wrong cell would be inefficient and could also cause health problems for the patient. Even once the right cell has been targeted, the gene has to be returned on. Avoiding the immune response, the cost of gene therapy. Examples: cancer. Several promising gene therapy treatments are under development for cancer. One, a modified version of the herpes has been shown to be effective against melanoma, a skin cancer that had spread throughout the body. The treatment, called TVEC, uses a virus that has been modified so that it will 1. Not cause cold sores, 2. Kill only cancer cells, not healthy ones, and 3. Make signals that attract the patient's own immune cells, helping them learn to recognize and fight cancer cells throughout the body. The virus is injected directly into the patient's tumors. It replicates, makes more of itself, inside the cancer cells until they burst, releasing more viruses that can infect additional cancer cells. Parkinson. Patients with Parkinson's disease gradually lose cells in the brain that produce the signaling molecule dopamine. As the disease advances, patients lose the ability to control their movements. A small group of patients with advanced Parkinson's disease were treated with a retroviral vector to introduce 3 genes into cells in a small area of the brain. These genes gave cells that don't normally make dopamine the ability to do so. After treatment, all of the patients in the trial had improved muscle control. Genetic engineering. Genetic engineering means the manipulation of organisms to make useful products, and it has broad applications. There are over 25 countries that grow genetic genetically engineered crops. The United States is responsible for producing almost half of the genetically engineering crops planted worldwide, and currently devotes over 40% of U.S. cropland to these modified crops. One of the most common types of genetic engineering is to insert the genes for bacteria into the crop. This type of genetic engineering works like an insecticide. The insects consume the crop, they will be infected by the bacteria, and eventually they die. The most noticeable benefit is that genetic engineering has made it possible to produce more crops in a shorter time of period, due to the modifications that make crops resistant to diseases, making possible to grow crops in regions that would otherwise be unsuitable for agriculture. Problems with genetic engineering. Insects will eventually develop genetic resistance to the chemical. Eventually, the genetically modified crops would no longer be successful. Other technique that uses genetic engineering is bioremediation. It's a technique in which biological waste expect some heavy contaminants. It's removed by using the metabolism from the organism. It can occur naturally or artificially. There are two ways in which genetic engineering can help to this process: by modifying a pre-existing microorganisms to make it absorb that waste and digest it, by designing brand new organisms to absorb the waste. An example of this is the Deinococcus radiodurans, a microorganism which eats radiation.